Alright, hey everyone, so this is Lab 10, pre-lab discussion and walkthrough. I want to thank Dr. Taylor and Jamie Gonzalez, the lab coordinator, for helping get all this together. So we ran into a couple hiccups. We were going to do a virtual lab with McGraw-Hill program, but it seems like we've lost that contract. We're no longer using NetText, so we don't have access to the course materials. That being said, you will use your lab manual, Lab 10 enzymes, to do this experiment. We will conduct the experiment and then provide pictures. You will go through and fill out the lab. But I'll get there in a little bit. First, let's look at the PowerPoints and do a little pre-lab discussion. Okay? So today's lab is enzymes, which you have seen a couple times already in this course, or in the last course and this course. Remember from the last course we talked about enzymes lower the activation energy to speed up reactions, and we talked about different factors that could affect enzyme activity. And then we revisited that in chapter 16 when we started to introduce enzymes and substrates and how they connect like a lock and key to facilitate these reactions. Okay, but we'll go back over all that. I was just going to say that you can go back in your text and find earlier sections that introduce the enzyme and talk about some of the more basic definitions for what an enzyme is. So the laboratory goals for today are to describe the role of an enzyme in biological systems, which we will talk about, set up chemical tests that measure the rate of an enzyme catalyzed reaction, observe the effects of enzyme concentration, temperature, pH, and inhibitors upon enzyme activity. Now, I'll look through the pictures. I'm not sure that we're testing all of that, but we will look at a lot of that. Okay, so what are enzymes? Enzymes are just essentially smaller proteins that facilitate these reactions. So if you remember from your text, they kind of describe it as like a lock and key mechanism. Okay, the enzyme is essentially a lock. Whatever substrate that needs to get in there is the key. They can only fit together in a certain kind of way, and they can only facilitate certain kind of reactions. Okay, so in that way, enzymes are very specific to certain molecules, can only connect, and then only one type of reaction essentially can proceed. All right, so the body has thousands, millions, maybe billions even of these little enzymes that help to facilitate all these little reactions throughout your body. Okay, whether it be digesting food, moving oxygen around, storing energy, moving energy, stuff like that, breaking down alcohol. All these different things require their own little enzyme to help the reaction go. If we didn't have these reactions, these processes might take weeks or months. So if it takes you months to break down the food that you eat, well, you'll be dead before you get the energy out of it, right? So that's why the body needs these catalysts to speed up the reaction, okay? So let's talk about how an enzyme does that. It does that by lowering the activation energy. So you've seen this chart before. Imagine this is a hill, essentially, that I have to push a rock over. When the enzyme is not present, I have to put a lot of energy into it to get the rock up the hill. All right. Now, once I get up there, it'll go and react just normally. But the difference in having an enzyme is not is the difference in a large amount of work. Right. I have to work a lot harder if the enzyme is not present. I also like to think about it like a bridge. Right. I think my iPad's down. It is. So when you're going, say I'm standing at a cliff and I'm looking across a ravine or a canyon to the other side, and I want to get across to the other side. If I don't have an enzyme, let's call it a bridge. The enzyme is like a bridge. If I don't have that bridge, I have to crawl down in the canyon, find my way, work my way all the way back up, and spend a whole lot of energy to get to the other side. But an enzyme is essentially just like a bridge. I can put this bridge down to the other side, and all I got to do is walk across. There's not a lot of energy being spent for me to get across to the other side. So that's kind of how you think of an enzyme. It's like building a bridge from one side to the other of a reaction, and it allows you to get across much more easily than if you had to crawl down and crawl all the way back up. Um, so that's what it does. It's called lowering the activation energy, and you see this graph here on the side. The one in purple is the one that was catalyzed, and therefore this energy of activation, this little arrow here, is smaller than the energy of activation for the one without the enzyme. Okay? So for this, we're going to test the enzyme activity of amylase. Real quick point, anything that ends in ACE, should signal to you that that is an enzyme. Okay, amylase is an enzyme that breaks down amylose, and typically they're named very simply like that. Hydrolase is something that hydrolyzes, an enzyme that hydrolyzes. So they're most usually they are named for the job that they perform. Okay, so you'll see ACE a lot dealing with enzymes. Every enzyme name ends in ASE. Okay, ACE. So today we're looking at amylase. Amylase is an enzyme that hydrolyzes amylose. Okay, and interestingly enough, it's essentially what's in your mouth. So when you say, for example, take a cracker and put it in a glass of water and let it sit and wait to see how long it takes for that cracker to break down into little pieces. Okay, a lot of people think when they're eating, that's essentially all they're doing is they're surrounding it with water and it starts to break down in your mouth just because of that. But you'll notice that putting it in your mouth will break down a lot faster than that cracker that you put into a glass of water. Okay, and the one in the glass of water might not ever break down fully, right? But when it's in your mouth, these amylases, the enzymes, are essentially speeding up the reaction. So they're breaking down all these starches and sugars and everything much faster than just a regular glass of water would do. Okay, and that's why they speed up reactions by hooking up to the sugar carbohydrate in a certain way, allowing it to break really quickly, and then move on to the next one. Okay, so it's just breaking down millions and millions of molecules like this. Pop, 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 pop. All right, so that's what's happening all throughout the body with all different type of things. The enzymes help to store fat. They help to break down fat when you need it, when you're using up all your energy and you have to go grab some of them fat cells. Enzyme will grab this huge fat molecule, start to break down little parts of it, send those out to the rest of the body for energy. Okay, so these enzymes are all throughout the body, and they function, and they help to function facilitate, I should say, a bunch of different reactions. All right. So yeah, we're going to look at amylase a little bit today. Here is a visual color reference. So once we start doing the test, you'll look for these colors to kind of indicate how much of the starch is remaining. So if you open this up in your own PowerPoint, you'll see down here on the bottom it's a gold color and it says none, enzyme activity 3. Okay, just in case you couldn't read that. Below light brown, there's another section called gold, none, and enzyme activity 3. I think if you open this up in your own PowerPoint, it'll fix that. I didn't notice it in this other one. Matter of fact, let me go here. That'll be better. Okay. Where are we at? Here. Okay, yeah. Gold, none, 3. Alright, so this is the color code for the experiment that we'll be conducting. Jamie did the experiment. She just provided the picture, so I have posted those on CougarView. You'll use those to answer your lab reports, but I'll talk about that more specifically here in a minute. Okay, so one thing that you were questioned on in chapter 16, and it was kind of introduced, was this thing called the enzyme substrate complex. And essentially, the best way to think about that is like a lock and key. Okay, the enzyme is the lock, the substrate is the key. They hook up in a very specific way. I can only put the key in one direction, right? It has to be straight up and down and going forward. So I kind of think about the enzyme substrate complex in that same way. That substrate has to be aligned in a specific way, and it connects to the enzyme in a very specific orientation. Okay? And then once they can connect, then the reaction can take place. The key turns and then breaks out, and maybe is something different when it comes off of the enzyme. Okay, maybe it's two parts that come off of the enzyme. Maybe it's just a different molecule. But all different kind of reactions can occur at that point. All right, so this is just a really simple illustration. Enzyme plus substrate leads to an enzyme substrate complex. Then the reaction occurs, the key turns, you have an enzyme product complex, and then the enzyme breaks away from the product, and you just have your new molecule or molecules, whatever. Okay? Um, so different things can affect the reaction rates of enzymes. Interesting. The body is actually a good illustration of how enzymes function and what specific conditions need to be met for them to perform properly. So, typically, human enzymes function at around 98 degrees optimally, right? You'll notice that's the body temperature. So all of these enzymes have kind of developed in a way that they work best at 98 degrees. Now we can get them to go a little faster, say by raising the temperature. All right, that's one factor that can affect enzyme activity, temperature. So this is like getting a fever, right? When you start to get a fever, it's telling these enzymes in your body to work harder, work faster, do more, right? And that's essentially how fevers work. They speed up all of these reactions. Sorry, I heard something going on. They speed up all of these reactions in the body up to a certain point. Now, they also function within a range. So, say enzymes stop working at 105 degrees Fahrenheit. All right. So that's what's inside the body. Your temperature is raising. It's getting faster. The enzymes are working more, 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 trying to get you better, trying to make you sick or pass through whatever is bothering the body. Right. That's what caused the temperature to go up in the first place. Something was bothering it, and then you had a response, told your body to raise the temperature. Enzymes started working harder. Once you got up to 105 or something, all of those enzymes stopped working entirely, and you died. Okay, so the point is the enzymes function between a certain temperature range. They have an optimal temperature at 98 in humans, and then you can speed it up a little bit, but you are to the detriment of other enzymes in the body or to other processes in the body, right? Other enzymes in the body ain't going to function as well at 102 degrees. But the ones that the body needs to get rid of this infection or whatever, they will function better at 102 more quickly. And then eventually, if it keeps going up 104, 105, everything breaks down and you die. Same thing on the other end. If it slows down too much, 92 or 93, something like that, then enzymes stop working, you die. So it's important that you kind of keep um, awareness of the conditions that the enzymes are in. Another condition that affects enzyme rate is pH. So here's the temperature, and I talked about the optimum temperature somewhere around there. I think that's like 98 degrees Celsius for the body, and then it falls off as it goes up. Denaturation, that's when everything breaks down. And then as it goes down, you see actually you can get a lot colder before everything stops working, as opposed to increase about 10 degrees Celsius, and it's over. All right, and so then I started to move on to pH. pH is works essentially the same way. The body operates at about 7 pH neutral, 6.7, 6.8 in the blood, something like that. So enzymes function most effectively at that pH. If you lower the pH or raise the pH, you'll see here this graph shows that the reaction rates start to decline. Okay, so that's how come all these conditions have to be maintained in the body. They call it homeostasis, right? You have to maintain a balanced pH. You have to maintain a certain water level. You have to maintain a certain temperature, a certain concentration of enzymes in order for all of these systems to function properly. And so a lot of this is where vitamins and different nutrients and stuff like that comes in. Certain uh, essential minerals, right? Vitamins and minerals, you hear that all the time. These things help to build up, number one, your enzyme capacity. The more you work these enzymes, the more efficient they become, right? It's kind of like an alcoholic, someone that becomes an alcoholic. They have certain systems in their body that break down alcohol. If I use that system more readily, it becomes more efficient at breaking down the alcohol, right? To the detriment of other systems. I'm putting a lot of energy into this liver to break down, process all this stuff, and it's getting better at it. But it's because I'm becoming an alcoholic, right? So all different kind of systems in the body use enzymes in the same exact ways, and they all are going through this little balancing act where we have to put energy into one thing. We have to choose how much energy we can afford to lose to that. Enzymes help reduce the energy cost. So that's how they work in the body. The effects of temperature, pH, enzyme concentration. More enzymes mean more reactions, right? That's kind of given. All right, all of them are the effects or factors that can affect enzyme activity. All right, temperature, pH, enzyme concentration. Yeah, that should be good for now. And inhibitors. Okay, so inhibitors are something that might bind to the enzyme or block the substrate from being able to attach to the enzyme. There are several different kind of inhibitors, and you'll see a list of them here. Just to mention a couple quickly, medications are typically used in this way, and we use them in every kind of way that you see here. Okay, competitive inhibitors, they bind to the same active site, but they also can release from that active site. So essentially, we're just kind of plugging up some of the enzymes to slow down the reaction. Okay, maybe we don't want to. Maybe that's something that's happening, say carbon monoxide poisoning, right? You've taken in some carbon monoxide. It's bound to where you typically absorb oxygen. And so now you can't take as much oxygen in. That's how you end up with carbon monoxide poisoning. Okay. Actually, carbon monoxide might be an irreversible inhibitor. I'm not real positive about that. I hadn't done much on that. But I'll come back to that and use it again there. Non-competitive inhibitors bind to something other than the lock portion of the enzyme. Remember I talked about the enzyme being a lock and key. Dang it. Am I still on me? Okay. I talked about the enzyme being a lock and key, right? Well, something else could bind to the backside of this lock and change the configuration where the key no longer fits. That's called a non-competitive inhibitor. And you'll see more illustrations of this in your text as well. An irreversible inhibitor. So let's go back to carbon monoxide. I want to say that's an irreversible one. So what it does is it will hook up to that lock portion of the enzyme and change the inner layout of the lock. Okay, so essentially it's like filling little spots inside the lock mechanism. Now my key would no longer fit, and not only that, but I can't get the carbon monoxide away from the enzyme. So this enzyme will not process oxygen at all. That's why we call it irreversible. This carbon monoxide is locked onto this enzyme and it's not letting it be used anymore. Okay? And so you'd have to process that enzyme and break it all the way down, rebuild another one just to get back your function of that particular enzyme. All right, and then reversible inhibitors are ones that can dissociate and restore activity. So essentially, they're not changing that lock there. All right. Now, all of these are kind of similar, but they have the same effect in that they just reduce the enzyme activity. Okay. Here are some non-competitive inhibitors. This is what I was talking about. It locks on to the back side. It comes on and attaches to the back side of the lock and changes the keyhole essentially. So now that substrate doesn't even fit in there. That's called a non-competitive inhibitor. Okay, I guess that's it for the pre-lab discussion. Like I said, none of this is stuff that you hadn't seen before. You'll be assigned a part to complete and share your results with the class. Share results, not answers. Don't worry about that. I am sharing my results with you. So when you go to Cougar View, make sure y'all can still see what I'm seeing. Looking through your lab manual, I had the same pre-lab discussion that we just did, right? And then you'll see a procedure. Let me switch over. Transition. Okay. You'll see a procedure starting on page 106, and this is what Jamie is essentially doing. Now, the only note I want to make about what you'll see here in Cougar View is these slides are upside down. Okay. I didn't realize it till it was too late, but I would start from the bottom and go up like this. Yeah. And even that doesn't work perfectly, but the first page you ought to probably read is this one because it tells you what to look for when you see the color changes. Remember, we're studying amylase and its ability to break down starches. Okay, just like it would in the mouth. So you'll use these images to answer your report sheet starting on page 111. Okay. So starting here, Enzymes, Lab 10, page 111, you should be able to do the time. Excuse me, let me go back here. This is the time, and you'll notice that it has all the colors there. The questions you shouldn't have any problems with. Let me look to part B. Part B is the effect of temperature. So here are the temperature tests, so to speak. And then you'll have some questions following up that. Next is the effect of pH. So that's back up here at the top, pH and enzyme. pH 2, 4, 7, and 10 are all conducted there. Now for part D, you'll have some follow-up questions there. And part D, we also have everything here. The inhibition of enzyme activity by what is that? Silver nitrate and ethanol. Okay? So, that's pretty much it. Email me if you have any questions about what's going on. And this is not due till the 27th of July. But if you want to turn it in earlier, get some feedback, I might can do that. So I'm going to work on grading the rest of this week. But this is the last lab that you have to complete, Lab 10, and that is it for the lab portion of the course until we get to the final. So Be looking for reviews on all the labs that we've covered so far, and a study guide for your exam. Okay? So that's it. I've enjoyed it. I will see y'all for the review.